Now, we've gotten through the first 3 steps. Step 1, formation of monomers. Step 2, formation of polymers. Step 3, formation of protective layer. Now we've got to get to the point where we get early cells. We get the cells taking on characteristics like reproduction, energy transference, and so on. This is going to occur through a process called chemical selection. Now, thus far in the course, what kind of selection have we talked about primarily? Natural selection. We're not there yet because we don't have that reproductive aspect. Chemical selection, based on the idea that one chemical is going to be built in large quantities if it is faster and easier than producing another chemical. That is, the easiest chemical to make is going to be the one that's predominantly made. Does that make sense? So in chemical selection, if you're making something and it is easier to produce, let's say it's easy to produce salt, harder to produce baking soda. As a reaction occurs that could produce either one of them, salt will be produced in much, much higher quantities. That's chemical selection. At this stage, we have big polymers. We got that in stage 2, right? We excluded water, we built polymers. Polymer of DNA, the polymer of RNA. We got the polymer of polypeptides. The most predominant polymer at the time was very likely RNA because it was chemically selected for. It's RNA that likely became the first macromolecule found in protobions. Based on what you remember from 101, what is the big difference between RNA and DNA?
DNA is deoxyribose and RNA is
ribose. Right. So RNA uses deoxyribose as a sugar. DNA uses deoxyribose. RNA uses ribose as its sugar. That's a big difference. The other difference you probably remember is DNA is double-stranded, RNA is single-stranded. And that's what we've taught you probably through most of your life, right? Do you guys remember that? Okay, good, because that's wrong. Turns out RNA can be and is oftentimes double-stranded, just not in the one process that you've learned it with. Nucleotide sequences are great at storing information. There are 4 possible bases, 5 if you include uracil, that alternate. And not only do they alternate, but they are able to be self-binding. So you can build one type of nucleic acid off of another. You guys learned a lot about that in Bio 101. You remember the whole transcription process, replication process? That property of nucleotides, that being able to bind to themselves, allows them to basically make an exact copy of its own strand, reproduction. The trick is figuring out a way to make it copy itself. Self-replication has to become key. Whatever early molecule in chemical selection has to be able to build itself. It's like a robot building a robot building a robot. And RNA, more than DNA, has the capacity for self-replication. RNA can build other RNA. And it occurs because of this structure here, which is known as a ribozyme. A ribozyme is a piece of RNA that's folded back on itself that acts as a catalyst to build more RNA. This we know exists currently, which means that at some point in the ancient past, it evolved. It happened. Now, what are the chances of it happening by sheer dumb random chance? Sounds pretty low. But once it happens one time, and all you need is one time, now you've got something that gives it a selective advantage. And you guys have seen with alleles and allelic frequencies, once you have a reproductive advantage, you begin to take over very, very quickly. So ribozymes are going to synthesize macromolecules. Yes?
Like mathematically, what are the odds that it happens first time?
That's what we're going to... Don't worry, I'll get into the math in just a second. But there is math involved. So over time, any RNA that picked up a mutation that allowed it to be more enzymatic, that allowed it to make this process happen faster, it would then increase that chemical selection that much more. That organ or that particular piece of RNA would have a chemical advantage and reproduce itself even faster. So very early in Earth's history, it was hypothesized that we had an RNA world. That RNA became encased in a liposome, maybe around clay somewhere. And once that RNA in that ribozyme form became encased in a liposome, it began to and could reproduce itself over and over and over again until it became the dominant proto-life. But like you said, what are the chances of that happening? How hard is this? Can we show that it's possible for this to occur? Questions so far, guys? Time goes on, the liposome acquired enzymatic ability to attach nucleotides, and the organism bigger. Slowly, as chemical selection acted on the RNA, it became able to bind amino acids to itself, or to each other. This began to build enzymes, and once you had enzymes and proteins, it became a slope. Evolution began to occur very, very quickly at that point because chemical selection would have favored these proteins that were even more efficient than ribozymes. So here's how we got the evidence. And this is tricky. This is Dr. Jack Szostak. He is a Nobel Prize winner. And I think he won the Nobel Prize for this. He also may have won the Nobel Prize for recombinant DNA technology. But what he did back in the 90s, he took 10 to the 15th power. So I'm going to write that down up here. So 10 to the 15th power is 4, 5. So that is 1 million, billion, trillion, one quadrillion RNA strands. And he mixed them up. And he mixed them with copies of short RNA strands. These were random RNA strands. These aren't anything from humans or anything like that. Basically, they were just lab created and completely randomized. He mixed them into a container together. Then he allowed selection to occur. What he did was he took he selected for long RNA strands. So if you started off with RNA that was this long and this long, he put them all together. He waited a bit, and then he selected for RNA that was longer than the first. Why did he select for RNA that was longer? Because it somehow added the short ones together. Does that make sense? So now he takes these long ones. He runs another experiment where he takes just those long ones. Again, this was a random sequence. He mixes those together, and he finds some that are now even longer. This is chemical selection just happening at a much faster pace. He did that again, longer and longer and longer. What he found was that those long RNAs were acting as catalysts to speed up this. He repeated the experiment 10 times, and only 10 times. After the 10th experiment, he found that RNA bonding efficiency was 3 millionfold faster. It was 3 million times better at binding nucleotides to each other than the original completely randomized bit of RNA was. He created an enzyme just through selection, or ribozyme just through selection. So what are the chances of it happening in nature? Pretty darn good. He got this to happen after 10 runs. Now there was he was doing extreme selection, but given the 3 billion years of Earth's history before we have these molecules really forming, that's plenty of time for this to have occurred. And likely it occurred shortly after the last bombardment cycle of meteors. So theoretically, RNA molecules can. So the question became, chemical selection's favoring RNA, so where'd the RNA world go? And that became all that stuff you learned about in Bio 101, where you're going to have RNA that then, or you have DNA, which is a very stable form of nucleic acid that promotes long-term energy storage. RNA is still useful in that process. What's RNA used for with transcription or translation? How about that? Building a protein. What do you use the RNA for? from Bio 101? It's the messenger. It takes the DNA message to the ribosome. But if we look at RNA, everyone's like, well, DNA builds RNA, not the other way around. RNA actually can build DNA. It builds DNA in viruses all the time. RNA-based viruses take a piece of RNA, they reverse transcribe it into DNA, and boom, you've got DNA that can now make proteins. The only benefit that DNA has is that it is more stable, but RNA does everything else. DNA provides that stable platform from which you can exist from generation to generation. RNA still does all the work. RNA does all the enzymatic function. So we can produce DNA from RNA. So don't need DNA for that. We can catalyze these reactions using RNA. What do you guys remember about RNA from Bio 101? What is it used for, aside from being a messenger? It does make proteins because it travels from DNA to the ribosome. But what does it do in that process? Right, so you're okay. So we'll go into it in a little bit more depth in just a second. Because we have DNA, and we use DNA for information storage, but what do we use as enzymes? Because we're not using RNA. What macromolecule are we using as enzymes? Proteins. So now we've got DNA that gives us a message, a way to build polypeptides. Now, peptides are composed of, polypeptides are made up of about 20 amino acids, each with their own unique variable R side chains. Those R side chains allow the protein to have a lot more different forms than RNA might have, a lot of different shapes, and with more forms come more functions. So then these polypeptides begin to get that chemical advantage. They're now able to do even more. They can link things together even better. So they're sort of building off what RNA originally did and going even farther. And that's where we're getting to today's world, where we have DNA as the storage, RNA as a transmission, polypeptides actually doing the work. But even today, all of this synthesis is still fully based on RNA, even if you may not remember it. Protein synthesis is entirely about RNA. So the big one that everyone remembers is messenger RNA. Does anybody not remember anything about messenger RNA? Like, messenger RNA is not new to you guys, right? It takes that information from DNA to the ribosome. But what you may not remember is all of the other RNA that's involved in this process. tRNA, transfer RNA. Do you guys remember that at all? Okay, now it's coming back. There was a lab. You matched things up. Transfer RNA carries an amino acid on the acceptor stem to the ribosome. So we've got messenger RNA carrying a message, tRNA carrying the individual subunits. But wait, there's more. Ribosomal RNA. In order to match up with the messenger RNA, the ribosome's actually composed about half to 60% is RNA of a ribosome. So ribosomes are made of RNA. You have a large ribosomal subunit and a small ribosomal subunit, each with their own distinct RNA subunits inside. So you've got lots of things going on where you're still, even in today's world, using RNA. It hasn't gone away. So that chemical selection in earlier history led to natural selection. And it ultimately led to the current central dogma of molecular biology, where DNA is transcribed to RNA, RNA is translated to proteins, and proteins allow replication to occur. That's where we are today. If we didn't start there, we likely started with a strand of RNA that was just super good at connecting to other RNA. Self-replication. Now, our current cells in today's modern world, bacteria and eukaryotes, they're more complex than those protobionts, because we have been undergoing round after round after round of natural selection. It's no longer a chemical selection phase. Which thing is the fastest to build? Now it's what information that's passed from one generation to the next is the most efficient at producing an organism. So we, in these past few lectures, have gone from non-life. We've gone from random chemicals to forming the monomers of organic molecules. Those monomers became polymerized. Those polymers became encased in a protective layer, and then that protective layer took on cellular characteristics. At that point, we get from a chemical selective phase to a simple life phase. These now protobionts are early cells that can reproduce and pass information from one generation to the next. That led to natural selection and the more complex life we see around us now. That is where we are currently. So, 4 big things: creation of monomers, creation of polymers, encasement of the polymers in a protective layer, and a chemical selective phase in which these protobionts take on the aspects of living organisms. Questions or concerns? Yes?
Can you give me the really embarrassing Bio 101 reminder of what is a monomer?
Absolutely. So what's a monomer versus what's a polymer? So if you remember from Bio, which you don't, but if you remember from Bio 101, you had individual subunits that would be created. So a monomer is, say, a sugar. And then you put those sugars together. Mono means one. So put those sugars together to form a polymer, which is many. So for, say, starch, you're going to use glucose, and then you will attach those glucose molecules end to end to end to end to end to get a polymer. So for a polypeptide, you have an amino acid that will bind to another amino acid that binds to another, to another, to another, to create a big long strand of a big long polypeptide. That's the polymer.
So wasn't it there's lipids?
Yes, so there's 4 classes of macromolecules: lipids, or fats, lipids, proteins, nucleic acids, and carbohydrates are the 4 large classes of macromolecules.
Nucleic acids are made of
Nucleic acids are made up of nucleotides.
polypeptides that are made of.
Polypeptides or proteins are made of amino acids.
Oh, okay. Gotcha.
Yep.
All right.
So yeah, you do kind of. That's why I do ask you guys to remember all that Bio 101 stuff, because it does actually come in really handy, especially this chemistry component, which was all. Sorry about giving you so much chemistry today. I promise I won't give you as much chemistry in the future all at once, at least.